Welcome back to botany class. In the previous class, let's discuss basic concept of plant tissue culture. Today you are going to learn very important question, that's technique involved in PTC. What's PTC? Plant tissue culture. What are the techniques involved in plant tissue culture? The techniques are sterilization, media preparation, culture condition, and induction of callus. Embryogenesis and hardening. These are the different techniques involved in plant tissue culture. What are the techniques? Sterilization, media preparation, culture condition, induction of callus, embryogenesis and hardening. What is sterilization? You know that this plant tissue culture, culturing the plant tissue, either a single cell or a tissue or a organ from a plant, it's derived from any part of the plant, and culture it in the artificial synthetic medium, aseptic condition, because it's the method of growing a plant in a small glass pot, glass tube or any glass ware with cultured medium. So this technique is very much important. Sterilization, all that is sterilized. So first step of this technique is sterilization. All the parts used for this technique, used for the culture, is sterilized. So at first aseptic environment can be produced. So what are the sterilization process? Sterilization, sterilization of the explant, sterilization of the medium, sterilization of the environment, the room, everything must be sterilized. So first sterilization is the maintenance of sterilized environment. The environment condition is sterilized. So the culture room, the area where it is selected to sterilize. So all the equipments used for the culturing plant, that including the forceps. What are the things necessary for this? To transfer this, to take the explant, sterilization of the explant. By taking the explant, it's not take it in our own hand. Forceps can be used. The forceps, scalpels, and what are the things? Glassware, glass vessels. These all must be sterilized by using wet steam, using autoclave. The glass vessels may be a test tube or conical flask or petri dish, anything. The glass wares, all the glass wares must be sterilized with autoclave. Autoclave using, there are 15 to 30 minutes by 15 psi temperature. So it provides, this autoclave gives the wet steam. So all these glassware, the forceps, scalpels, glass vessels, what are the things necessary for this? These all tools can be sterilized by using wet steam by autoclave 15 to 30 minutes. So, this is surface sterilization, the maintenance of the environment. The environmental conditions must be sterilized. What is sterilization? The definition of sterilization is get rid of microbes from the environment. Get rid of microbes. There are a number of microbes in the form of bacteria or fungi. So, this bacteria or fungi can be removed from the area where this plant cell is going to culture. So, sterilization is the process by which get rid of microbes from the area where you are selected for culture medium, either in the lab or in the room. That must be sterilized. So, first sterilization, produce the atmospheric condition, the environment, maintenance of the environment, sterilization. Second one, the sterilization of the nutrient medium. Second sterilization process is nutrient medium. Nutrient medium. The nutrient medium is a liquid. So, it must be sterilized by using either using some alcohol, or that is 70% ethanol can be taken and sterilized, or it is used by autoclave, the wet steam sterilization. Autoclave 15 to 30 minutes the sterilization given to this. So, medium also sterilized. The medium may be used, there are different kinds of nutrient medium can be used. The nutrient medium in the form of MS medium, Murashid and Skoog medium called as MS medium, or another alternative medium is G5 medium, that is Gamberg medium or B5 medium or white medium. The common medium which is used is NITS medium. So, Murashid and Skoog medium, G5 medium, white medium, or NITS and NITS. NITS medium can be used. So, MS medium, and this can be added with the vitamins or hormones or the plant extract. The plant extract can be passed through by the filter 2 mm. Filter it and add it to the nutrient medium with agar. Agar is a substance, solidifying substance. So, the agar, the preparation of this, making this medium can be solid or semi-solid or liquid. It can be solidified with agar medium. So, agar is prepared from a marine alga. So, this medium is used for solidification. So, it used in the some state, solid state, either the liquid or gas or the semi-solid, the solid or liquid or any forms can be solidified with agar. So, it also added to the medium. So, nutrient medium can be sterilized. And the explant, sterilization of the explant. Before sterilizing the explant, the room can be sterilized. Sterilization of the room. In the room, the floor and the walls can be sterilized with washing with detergents. At first, wash with detergent, and it must add 95% ethanol and exposing UV radiations to sterilize the room. And explant must be sterilized. The explant, what is explant? The plant part which is selected. Here the explant is selected as leaf. Here leaf is the explant. The leaf must be sterilized with running water, tap water. In the tap water we wash the debris which can remove. Surface sterilization. Then 0.2% mercury chloride for surface sterilization. Finally, this ethanol can be used for sterilization. So, this all sterilization is very much important. So, what sterilization? Sterilization is get rid of microbes like bacteria and fungus from this tissues or in the room or in the environment. Make this environment aseptic. To produce the aseptic environment, the maintenance of sterilization in the environment and in the nutrient medium and the explant as well as the room. So, first the environment conditions, the tools, everything can be sterilized, then the room must be sterilized and the explant must be sterilized and the nutrient medium must be sterilized. And this all tools are used and the work is done in front of the laminar airflow chamber. Laminar airflow chamber. The laminar airflow chamber or laminar airflow bench. Here the air can be moved in a same direction. So, without allowing any cross air to getting microbes. So, it maintain as a aseptic conditions. So, at first sterilization, first process is sterilization. So, after sterilization, second process is preparation of media. The preparation of media. So, this preparation of media, here there are different media are used for culturing. Look at this diagram. This is the explant. Explant is the part of the plant. It may be selected the root, stem or leaf. Here it is selected as the leaf and it must be sterilized, surface sterilization, and selected a tissue. This is the tissue sample. And this tissue sample can be transferred to a nutrient medium. The media preparation. So, preparation of the media, when the cells grow in a culture medium, it cannot grow naturally. So, it is taken into a culture medium. So, here is the culture media which contains hormones like auxin. Auxin is the growth promoter. So, auxin and plant growth regulators. This all nutrient medium which is maintained by pH, temperature. It must maintain the pH, the temperature and light intensity, everything. So, in the media preparation, it must be taken as Murashij and Skoog medium, either this Murashij and Skoog medium or this Gamberg medium or white medium, nits and nits medium. So, at first it should be sterilized with the autoclave. So, allow it to 15 to 30 minutes. It allow in the autoclave. So, after sterilization, this explant is passed to the or transfer. This tissue is called as inoculum. Transfer of explant into the culture medium is called inoculation. So, inoculation is transfer of explant. Now, it is transferred to the medium. What are the medium? There are Murashij and Skoog medium, G5 medium. One medium is not sufficient to develop the entire plantlet. So, depend upon the plant species and depend upon this, it can be selected. Commonly selected medium is this MS medium, Murashij and Skoog medium. Other alternatives are G5 medium or white medium can be selected. Then culture condition. So, it cannot grow when the explant is transfer it into the nutrient medium. The culture conditions of the culture is very important. The culture conditions are pH, temperature and light. Normally when the plant grow, it exposure of day and night, equal day length and the night. Here also the pH, the normal pH, it is maintained 5.6 to 6. The normal optimum pH is 5.6 to 6. And the temperature and humidity, the humidity is humidity it has to given 50 to 60% humidity, 50 to 60%, 60% humidity. and the light intensity, there are approximately 1000 lux. 1000 lux light intensity. So, pH, temperature and light intensity, and the photo period 8 to 16 hours. Photo period. The photo period is 16 hours. So, this all comes under the culture condition. In this condition, this explant or this inoculum which develops, induction, next process is induction of callus. Induction of callus. So, first process sterilization, next media preparation, medium can be prepared, it is ready to transfer the tissue into the medium, and the culture condition, maintaining all this pH, temperature, light as well as this photo period. Now, induction of callus. Look at this diagram, here the callus, this is the callus. Callus is the undifferentiated mass of unorganized tissue. So, from a single cell or a few cell selected from the leaf, here the part of the plant which is taken as leaf. From the leaf there are few tissues. In this, it started to develop a undifferentiated mass of cells inside this nutrient medium. It produces, it divides repeatedly because it also contains auxin. So, it promotes division and promote growth. So, the cell divides and produces a mass of tissue called callus. So, callus is the undifferentiated mass of unorganized tissue. It will develop inside. So, inside this nutrient medium, callus develops, induction of callus. So, before that the process is inoculation. What is meant by inoculation? Transfer of the tissue into the nutrient medium. The tissue may be root or stem or leaf or tuber or any part of the plant. It can be transferred. So, for transferring this, in the sterilization process itself, they are using the forceps, scalpels, everything, or knife. It can be cooled, flamed and cooled. So, after surface sterilization with alcohols or 0.2% mercury chloride or 70% ethanol or 95% ethanol, if they surface sterilize, then it is shown in the flame. So, flamed and cooled forceps can be used for transfer these tissues. These all work under laminar airflow chamber. So, it is maintenance of the aseptic environment in the laminar airflow chamber. So, in front of this laminar airflow bench, this all transfer of materials can be done. Then, induction of callus. Now, callus induced. Then, next process is called as embryogenesis. So, after induction of callus, here the mass of tissues can be developed. Now, the nutrient medium can be changed depend upon the species, different species. The nutrient medium can be changed. Now, what are the additional nutrient medium need? Everything should be added for promoting further growth, differentiation, cell differentiation. This callus separated from the single cell and cultured. So, here callus is separated. This from here number of mass of cells are there. These cells are separated and it is cultured in different nutrient medium. Further division, it is allowed to divide further. Differentiation takes place. So, callus separated. Here callus is the mass of unorganized and undifferentiated cells. So, just a mass of tissue is developed. It can be separate and it is cultured in nutrient medium. A single cell culture. Each cell can be cultured to multiply the number of the plantlets. That is called embryogenesis. So, what is the embryogenesis? The differentiated cells now further divide and it produces the embryo. Here the embryo is somatic embryo, it develops. Somatic embryo. Now this embryo is named as embryoid. The name of the embryo is embryoid because it's the somatic embryo. The name of the somatic embryo is embryoid. So here there is a formation of embryoid, embryogenesis. Somatic embryo, development of somatic embryo is formed. The last technique or last process is hardening. Now these all tissues are developed inside the glass vessels or glass vase. So within the test tube or conical flask or a petri dish, it develops very small amount of nutrient medium, very small space. Normally the plant will grow in the outside environment, in the soil, exposure of direct sunlight. But all these conditions, natural conditions are provided artificially within an artificial room. Within the room and within the small vessel. So this plantlet can be developed in the test tube. The regenerated new plantlet. Now till embryogenesis, from this single cell is allowed to culture in different nutrient medium. From there it produces the plantlet, regenerated new plantlet. Now new plantlet can be produced. There are thousands of new plantlets can be developed. But within this test tube or this conical flask or petri dish, not sufficient to develop the entire plant. All the organs are developed, the root, the stem and the leaf part will be developed, but it can be allowed to grow. So final process is called as hardening. What's this? Hardening is the plantlets, the developed plantlets are transferred stepwise to the direct environment. So at first it's transferred to the greenhouse. Greenhouse is that's made up of glass. The roof and the walls are made with glass. So this house it can be transferred, or then it is transferred to small pot, that is hardening. So it's the process by which small stepwise reaction, stepwise process to transfer this plantlet into direct environmental condition, exposure of normal condition. So till the plantlet develop, it is inside the room. So artificially it grown. Now it can be transferred to the natural environment. Such process is called as hardening. So hardening is transfer of plantlet to the direct environmental condition. So these are the steps involved. The question is techniques involved in plant tissue culture. What are the techniques? First technique is sterilization. Sterilization, maintenance of aseptic environment. Aseptic environment first creates in the room, the laboratory can be created as a aseptic condition. Then media preparation, the media, there are different nutrient medium, it can be sterilized with autoclave. And the culture condition, pH, temperature, light intensity and everything must be optimum. And induction, so allowed to produce induction. So this temperature, inoculation and this inoculation is transfer of this X plant. Incubation, this two word is important. Inoculation, one is inoculation, another one incubation. What's meant by inoculation? Inoculation is transfer of this X plant to nutrient medium. But incubation is providing temperature. This temperature, maintenance of likewise like this pH, humidity, light and photo period. This temperature is 25 to 25 degrees Celsius plus or minus 2 degrees Celsius. This temperature must be provided to produce the induction of callus. To induce callus formation, this much temperature, humidity, pH, photo period and light intensity must be provided. And next process is embryogenesis and final process is hardening. Now another question is, what are the other types of plant tissue culture? There are different types of plant tissue culture. Depend upon the part selected from the plant tissue culture type is based on the explant. So these all parts, either leaf or stem or root or embryo or anther, any part of the plant. So it depend upon this explant, the plant tissue culture types or varies. What are the different types? They are the different types of plant tissue culture. Organ culture, number 2, meristematic culture. Here one example, meristem culture. Third type is protoplast culture, and cell suspension culture. These are types. So first question is techniques involved in plant tissue culture. Second question, types of plant tissue culture. What are the types of plant tissue culture? This type is based on the explant. Based on explant, the plant tissue culture is classified into organ culture, meristem culture, protoplast culture and cell suspension. What's meant by organ? Organ culture. Organ. What's organ? Look at this. Here there is a plantlet. This plantlet contains, at the time of regeneration, this callus undergo, when the callus is separated and make it to develop into the entire plant, different organs can be differentiated from the cell. The root, the stem, the leaf, all these parts are separated. That is a regeneration or formation of embryogenesis. So embryo, somatic embryo developed. From this embryo, organs are developed. For this culture, organ culture, we may select any part of the organ, any organ, like this. Here the explant is leaf, or the stem, or the root, or the tuber, or embryo, or anther, or ovaries. So any part of the organ. So these all are organs, different organs. Any organs can be selected and it's allowed to culture under tissue culture technique. That's called organ culture. The second one, meristem culture. Look at this diagram. This is the example of meristem culture. What's meristem? Here the meristematic cell is selected, either the shoot apical meristem or the root apical meristem. Here the example is meristem culture. So here is the leaf part. The older leaves are removed. Here this is the meristematic region. This meristematic region is selected. So older leaves are, by cutting, the older leaves are removed from this shoot, and only this part is selected for culturing. So this is the shoot tip. They are selecting the shoot tip with growing tip after remove the enclosing leaves. This twig is selected, removed all this enclosed leaf and select only the shoot tip. This is the shoot meristem culture. Shoot tip only allowed to grow. So this is the diagram showing magnified view of shoot tip. Only this shoot tip is magnified. After remaining part of the larger leaves can be removed and selected only this part, shoot. Meristematic region, the young region, meristematic shoot, the tip. Usually the tip region contains meristematic tissues. So meristem can be derived and it is allowed to culture. So now it is transferred to culture medium. This is the culture medium. The meristematic tissue is transferred. So inoculation. Here the process is inoculation, transfer of explant to the culture medium. Inoculation. So when it is make it into the culture medium, when it is transferred or it is provided to the culture medium, it is distributed to the culture medium, this callus is induced, induction of callus. So this is callus formation. Finally, from this callus, embryogenesis, that is organogenesis, and it developed the new plantlet. So new plantlet is developed under meristem culture. Then in protoplast culture, what happens? The protoplast can be removed. What is protoplast? In the cell, this is the plant cell which contains the cell wall. Inside the nucleus, the endoplasmic reticulum, chloroplast, the mitochondria, all the cell organelles in eukaryote, all the cell organelles are present inside. When you remove this cell wall, what is present inside? This is called protoplast. So, isolation of this protoplast and allow it to the culture medium. So, this process is called as protoplast culture. So, in the next class, we are going to learn protoplast culture and cell suspension. So, today the question you are going to learn is 5-mark question, very important question. Technique involved in plant tissue culture. The only 6 steps are there. What are the different steps? Review this: sterilization, media preparation, culture condition, induction of callus, embryogenesis and hardening. So, learn this question, draw this diagram in the class note, and this is example for meristem culture. Draw this diagram. These 2 diagram you draw it in the class note, maintain the class note and learn this question. Daily question, prepare well. Thank you.